So Oliver Hsia is a postdoctoral cell biologist in Alessio Cully lab. He completed his PhD in Timo Kurat's lab at the University of Glasgow, where he characterized the role of the NET8 E3 ligase DCNL5 in the apoptotic pathway. Matthias Hinterndorfer leads functional genomics characterization at Georg Winter's lab at CeMM. He earned his PhD in Johannes Zauber lab, where he discovered the ARKIN2 required for the import of proteasome 2 nucleus. And finally, Angus Cowan is a structural biologist in Alessio Cully lab. He obtained his PhD in Melbourne under the supervision of Peter Chabotar and Peter Coleman, where he studied the structure and function of pro-apoptotic BCL2 family proteins. The seminar is entitled An Intramolecular Bivalent Degrader Glues, an intrinsic BRD4 DCAF16 interaction.
Thank you very much, Nikolai, for the introduction. I just wanted to start by echoing the previous speakers and thanking the organizers and the DFCI for the invitation to share our story here. I think this webinar series is a really great learning resource and a really great interactive platform for the TPD community. So I just wanted to start going right back to basics, and I think this will be familiar to a lot of the viewers and the audience, but the TPD field kind of boils down to two kind of main modalities. You have heterobivalent PROTACs and you have monovalent glues, which includes molecular glues. And while there are fairly clear definitions for both categories, I think we would argue that the lines are reasonably blurred. And I hope that by the end of this talk, we can further blur those lines, but also maybe introduce another sort of modality in the induced proximity field. So it's often said, particularly in the TPD field, that BRD4 is an easy target and it's everyone's favorite target to degrade in PROTAC studies in particular. But in actual fact, it's everyone's second favorite target, at least according to the PROTAC database curated here by Wang et al., which is pretty comprehensive. And it's the second most popular target after AR, with over 220 PROTACs reported in the literature. And shown on the left here are a couple of examples, DB6 and MZ1. And this database also quite nicely highlights just quite how popular VHL and cereblon are as ligases recruited in PROTACs in the literature, with about 95% of these PROTACs utilizing one of the two. And this sort of disparity is further highlighted in the clinic, where I think all but one or two of the clinical candidates recruit cereblon, and the other one or two of VHL. And this is important because in preclinical settings, there is the emergence of resistant mechanisms, and so expanding the repertoire of ligases is increasingly important. And on that subject, aryl sulfonamides are quite an interesting family of compounds. They've been around since, I think, the 90s, discovered initially by Hsia, but it wasn't until 2017 that their mechanism was unraveled, where indisilam and some of the other derivatives were shown to be molecular glues, and they degrade the mRNA splice factor RBM39 by the recruitment of the COL4 substrate receptor DCAF15. And a couple of years later, the ternary structures were solved. Shown here is one of the examples of structure DCAF15, aryl sulfonamides, including indisilam and E7820 and RBM39. And despite the structural insight gained there really hasn't been much progress in leveraging these aryl sulfonamides as handles for PROTAC development. So shown here are a couple of examples. On the left is an in-house example of a PROTAC, and another one for the literature on the right. And the long story short is these aryl sulfonamide-based DCAF15 recruiting PROTACs are pretty rubbish. There aren't really any successful compounds reported to date in the literature. So as you can imagine, we were quite surprised when we came across this compound in the patent, which had a reported sort of BRD4 degradation potency in the 100 nanomolar range. And looking at it, it's 100 picomolar, and looking at it, it is a nominal BRD4 DCAF15 PROTAC. So you have the pan-BET inhibitor JQ1 as the BRD4 ligand. You have a short rigid linker. And then on the right-hand side, you have E7820, which is an indisilam derivative, and aryl sulfonamide, which is the DCAF15 ligand. And, we were interested in this, so we synthesized it and tested it in-house. And the degradation potency was as described. It was really, really potent. But what was striking and surprising was that we saw this level of degradation in a DCAF15 knockout cell line. So this was kind of the first of several surprises in the story of this compound one, as we term it. And in order to sort of follow up on this and put it through a bit of quality control, just to see what it was actually doing, we wanted to see if it degrading via the proteasome, via methylation, sort of the usual sort of PROTAC machinery. And to do this, we engineered an endogenous high-bit BRD4 cell line, and we're showing here kinetic degradation. And what you can see with the sort of purple and blue curves is rapid and sustained degradation in response to compound 1 treatment. But when you use the methylation inhibitor MLN4924 to block that, you block degradation entirely. And so that also is proteasome dependent as well. We're not showing here. And switching gear now to look at ubiquitination. So this is a kinetic ubiquitination assay. You can see, a strong ubiquitination signal in response to the compound. Again, this is blocked by the methylation inhibitor. But when we use the respective ligands that make up compound 1, so JQ1 and E7820, what you'll see is that JQ1 does block degradation, but when we do competition with E7820, the expected ligase ligand, we fail to block, ubiquitination, degradation, and the activity of the compound, which was the second main surprise in this compound. And to further interrogate this sort of atypical mechanism, we synthesized aryl sulfonamide containing truncations of compound 1, so essentially chopping from the left-hand side of the molecule shown here on the left, and we then used these in ligand competition assays. So we pre-treated with excess concentration of these truncates, and then added our compound 1. And what you can see on the right here is that only JQ1 blocks the degradation as before, but when you do E7820 or any of these truncates, you're still seeing complete BRD4 degradation, which is inconsistent with a PROTAC-like mechanism and suggests something else is going on here. We were then interested in whether the aryl sulfonamide moiety of compound 1 is actually required at all. And to that end, we synthesized truncations, this time from the right-hand side of the compound, from the aryl sulfonamide end. And what you can see here is that all of these truncations failed to degrade BRD4. None of them are active. And this suggests the aryl sulfonamide portion is absolutely required for the degradation activity, but in a manner that does not coordinate DK15. And the fact that we can't compete with the ligase end or the right-hand side of the compound suggests that it's not a PROTAC at all. And this was reminiscent of the GNE0011 compound, which at the time, all that was known about it was that it was a monovalent BRD4 degrader, which is also JQ1 based, and has been touched on by the previous speakers, and the ligase in the machinery was initially, I think, reported by his colleagues at AbbVie. And next, we were then interested in the target specificity of compound 1. And so we addressed this in a couple of ways. On the left, we did some TMT-based quantitative proteomics, where you can see, BRD4 degradation and BRD2 degradation, but no BRD3 degradation here, in contrast to, the pan-BET degrader DBET6. And when we engineered endogenous BRD2, BRD3, and BRD4 high-bit tagged cell lines, you can see again a strong preference for BRD4, so moderate BRD2 degradation and little to no BRD3 degradation, except at high concentration. And the next missing piece from the puzzle was then to look at the machinery. And for that, I'm going to hand over now to Matthias.
Yeah, thank you, Oliver. So with all of this in mind, and especially remembering that even though the aryl sulfonamide wide is supposed to target DK15, but the degradation is completely independent of DK15. The next obvious question was, of course, what is the cellular machinery that is engaged by this compound? And I'm not sure if you can see my mouse here. If not, maybe Oliver, you can show the audience the important part here. As the previous speakers already introduced, we also turned to a cellular dual fluorescent reporter assay to basically study the genetic components of this degradation machinery. And similar to them, we also fused BRD4 to a fluorescent marker, in our case BFP, and then used mCherry as a normalization and as a indication of specificity. And in these reporter assays, you can nicely see that the addition of compound 1 really leads to a potent degradation of BRD4 and of the BFP signal, whereas mCherry is completely untouched. So we could then use this reporter assay in a FACS-based CRISPR screen and combined this with the inducible Cas9 system. So we delivered the reporter to this KPM7 cell line that has an inducible Cas9 allele, then transduced these reporter cells with an machinery library targeting all known Calin ring ligases, then induced doxycycline to induce Cas9 expression, and then treated the cells with our degraders and sorted out the fraction of cells that are resistant to degradation induced by compound 1. Now I'm going to show you the results of these screens in these kinds of volcano plots, where always on the right-hand side of the plot, you can see the gene knockouts that mediate resistance to a specific degrader. And to validate our approach, I first want to show you the results of a screen performed with this well-established protoactive 6. And as expected, we can really recapitulate in these screens the known machinery that is engaged by DB6. So the Calin-4 complex, including the adapter proteins DDB1 and RBX1, but most importantly, the substrate receptor cereblon. Now, how do these screens look like with our actual test compounds? I would say remarkably similar. So again, we can find both with GNE and with our compound 1, we can find the whole Calin-4, DDB1, RBX1 machinery. Only this time, the top hit and substrate receptor that we identified here is not cereblon, but DCAF16. We could then, of course, also validate this dependency of DCAF16, both on the left-hand side in our reporter assay, but also importantly on endogenous protein, where we can see that in DCAF16 knockout cells, the degradation by GNE, but also by compound 1 are completely disturbed, completely perturbed. So, so yes, DCAF16 indeed is the ligase we were looking for. Now, you might be wondering, this was compound 1 was initially thought to be a DCAF15 engaging compound, but now we find it to be dependent on DCAF16. DCAF15, DCAF16 sounds so similar. What's the difference? You might already remember from the previous talk that DCAF16, even though it belongs to the same protein family, of course, as DCAF15, it is structurally completely different and unique. And I think we can nicely highlight this in this fluorescent polarization assay, where we can, of course, recapitulate that a FITC-labeled aryl sulfonamide probe nicely binds to DCAF15 in a dose-dependent manner, as expected, but it shows no interaction whatsoever with DCAF16. So we have this strange conundrum where our compound is dependent on a ligase that it obviously doesn't even bind to, at least in isolation. So we turn to a panel of biochemical and biophysical assays to better understand this mode of degradation. So firstly, here in size exclusion chromatography, when we mix BD4 with DCAF16 and with compound 1, we can nicely see in the blue track here the formation of these higher molecular weight species, so indicating that in the presence of compound 1, these two proteins form a complex and interact with each other. But the real surprise came when we performed the same assay, but this time left out the compound 1 degrader and found that the two proteins similarly interact even in the absence of a degrader. So speaking that for an intrinsic affinity of DCAF16 to BRD4. And importantly, so in the cyan track, you can see that no such interaction happens if we, instead of using a intact BRD4, we split the two bromodomains of BRD4 up and use isolated bromodomains in conjunction with DCAF16 and compound 1. And we observe no such interaction whatsoever with isolated bromodomains. So indicating that this complex formation and this interaction really requires the presence of both bromodomains at the same time on the same polypeptide chain. We then went on to perform an alpha lyso displacement assay, where we basically measure the ability of our compound 1 to displace a JQ1 based probe from BRD4. And we can see that in the presence of DCAF16, the affinity of compound 1 to BRD4 is dramatically increased. So speaking again for a strong trimeric complex formation. But again, this complex is only formed in the presence of both bromodomains at the same time, whereas no complex no cooperativity at all is observed when using only isolated bromodomains instead. And the final piece of evidence in this TR-FRET assay, we could again here in purple find a dose-dependent interaction of DCAF16 and BRD4, even in the absence of any degrader. So an intrinsic affinity of the two proteins to each other. Again, this complex is stabilized and enhanced in the presence of a degrader. And again, no such complex is formed at all if only one of the two bromodomains is present on BD4. So concluding that we have an intrinsic affinity between DCAF16 and BD4 that is stabilized and thereby turned or utilized by our compound 1 degrader, and B, that this degradation happens only in the presence of both bromodomains of BD4. This is then further corroborated in these cellular assays, where again we turned back to our dual fluorescence reporter and wanted to find basically the minimal degron on BD4 that is degraded by our compounds. So we chopped up the BD4 protein, and by removing one by one all of the protein domains outside of the two bromo domains, and saw that basically all of these protein regions didn't make any difference for degradation. So as long as we have both bromo domains present, the degradation is still very, very potent. However, if we go one step further and repeat the same assay just using isolated bromodomains, again, we could see that there's absolutely no degradation happening both with compound 1 or with GNE, whereas most of the established BD4 degraders here exemplified by DBAT6, of course, are very potent degraders even of isolated bromodomains. Next, you will hopefully remember from Ollie's part that in our TMT as mass spec assay, we had found potent degradation of BRD4 and BRD2, but no degradation of BRD3 at all. And so again, we wanted to make use of this reporter assay to basically dissect this specificity for BRD2 and BRD4 over BRD3. Again, this specificity is recapitulated here in our reporter assay. And so we could then go one step further and basically make hybrids of the two of these proteins by exchanging one part of BRD4 with the respective counterpart in BRD2 or BRD3. So what's not shown here, but what we did is firstly replacing the linker region that connects the two bromodomains to each other, and we saw absolutely no difference. So indicating that the linker region doesn't have anything to do with the specificity. Similar is true when we replace the first bromodomain of BRD4 with the respective domains of the other paralogs, and again we saw potent degradation. However, if we then turn to the second bromodomain and replaced the BD2 from BD4 with the counterpart from BRD3, we observed no degradation whatsoever with compound 1, and to a lesser extent, even GNE-based degradation was resistant now. So indicating that really the second bromodomain mediates this specificity for BRD4 and BRD2 over BRD3. And in the final part of the presentation, Angus is now going to show you how he used structure and cryo-EM to further dissect this specificity and the mode of action of our compound 1.
Great, thanks, Matthias. So I'm pretty much going to dive straight into the structure. Briefly, in order to get this reconstruction, what we did was take a tandem bromodomain construct, which consisted of the BD1 and BD2 linked by the native linker sequence. We mixed that with compound 1 and DCAF16 in complex with DDB1 and DDA1. We put that onto grids and collected a cryo-EM single particle dataset on our Glacios microscope here in Dundee. And this is the strategy we took. Some of you will have seen this in the preprint to get the reconstruction at about 4 angstrom resolution. I can briefly say since then, with further particle creation, we've managed to improve the resolution to about 3.8 angstrom. We've also just improved the quality of the map significantly, such that we can now build an atomic model with all components in the ternary complex. And this is what that ternary complex looks like. So at the top here, you can see both bromodomains of BRD4. They're bound to compound 1, which sits between the bromodomains and DCAF16, and the adaptive protein DDB1 at the bottom. And the real revelation from this structure, something we completely were not expecting, is that there's only a single copy of this compound 1 bound to the bromodomains. We'd been expecting up until this point, working on the assumption basically that JQ1, 2 copies of this compound bound to BD1 and BD2 respectively, and glued them to decaf16, but in fact, that's not what we saw. I'm a crystallographer by training. This is my first cryo-EM structure, and I'm really glad I'm getting to present it around Easter, so I can say Happy Easter to everyone who celebrates from my little cryo bunny. And given this is a double billing on decaf16, I thought it would be nice to take a quick look at and compare the two structures, particularly the position of BD2, which is present in both. You can see here that they look quite similar, and indeed the BD2 position is pretty much identical in both structures, which is nice cross-validation of both of these. But now to the key observation from our structure that I mentioned earlier. And this key observation was that compound 1 engages BRD4 bivalently and glues it to decaf16. So on the left-hand side here, you can see JQ1 bound to the canonical acetyl-lysine binding pocket of BD2. It and the linker sit between BD2 and decaf16, while the E7820 moiety surprisingly bound into the same acetyl-lysine pocket on BD1. You can see down at the bottom here some of the residues involved in this interaction. I've also just highlighted C58, which was the residue responsible for the activity of the covalent compounds. And I can say that in relation to JQ1, it sits in a very similar position in both structures as well. Interestingly, we've kind of found after the fact that actually there are some structurally distinct aryl sulfonamide compounds that don't contain this indole that have been reported as BET bromodomain binders in the literature. These were both reported around 2011, 2012. And we found that in an alpha lyser displacement assay with both BD1 and BD2, we could weakly displace the biotinylated JQ1 probe using E7820 and indicilam and some of our sulfonamide containing truncates. So beyond the revelation of the bivalency, the structure explained a lot of things that we'd observed already. So Matthias mentioned that there was, and Ollie has said as well, that there was selectivity for BRD2 and BRD4, and this selectivity was centered around BD2 of BRD3. So we managed to narrow this down to a single residue change in BRD4 and BRD2. BD2, this glycine 386 sits at the interface with decaf16 with these residues here. If you even sort of conservatively change this to an alanine, you're starting to get slight clashes with the decaf16. And you can imagine this residue is a glutamate in BRD3. If you change it to this bulky acidic residue, you're going to significantly disrupt this interface. And indeed, when Matthias put this single mutation into his reporter construct for BRD4, we saw the same thing as we saw completely replacing BD2, in that you completely abrogate degradation by compound 1 with this mutation. And I think the ternary interface is really quite exquisite. There's a number of interactions that define it. The one I just mentioned, the BD2ZA loop engages decaf16. We also saw that hydrophobic residues of decaf16 enclose the JQ1 moiety of compound 1. For example, this tryptophan sits against this dimethylthiophene ring, which would be exposed to solvent in the binary structure with just BD2. We also saw this tryptophan from decaf16 sort of sits in this little shallow hydrophobic pocket on the BD1. And beyond interactions between decaf16 and BRD4, we saw interbromodomain interactions. So the WPF shelf is a conserved structural motif in BET bromodomains. You can see here that the WPF shelves of BD2 and BD1 run roughly perpendicular to each other, and sandwiched between the tryptophan of BD1 and the proline of BD2 is this BD2 residue methionine 442, which also contacts compound 1 in the ternary structure. So this slide really is to highlight the fact that we're dealing with something new here. This is neither a bivalent PROTAC nor a monovalent glue. I've picked a couple of examples that I think are relevant here of bivalent PROTAC MZ1, which forms a ternary complex between BHL and BRD4, BD2 in this case. and the monovalent glue, of course, E7820, which glues RBM39 to DGAM15. And I think it's interesting to look at the buried surface areas in these ternary complexes between the different components. So if you look at the protein-protein interaction, you can see here that we're closer to the value seen for the glue system in the interaction, the smaller interface of the compound and protein. You can see where sort of similar to the glue, but a little bit larger. But in the larger interface between the compound and one protein partner, you can see we're at a very high value here because of this bivalent engagement of the same protein. And the overall interaction, buried surface area, is quite large, probably going some way to explaining the very high potency of this compound in cells. So I think we have a new modality on our hands here, which we're really excited about. So it's distinct from intermolecularly bivalent PROTACs. It's distinct from monovalent glues. It kind of has some aspects of both of them, but involves binding to two domains on the same protein and gluing it to a third one. And I think that this has relevance beyond just TPD, but in chemically induced proximity in general. And so just to kind of give a bit of an outlook of what we think this will mean for the field, I think it's an important cautionary tale in using weak ligands for PROTAC development, and shows the importance of using ligase knockouts and control compounds when developing any PROTACs. I think in terms of the general applicability of this bivalent gluing type approach, it's interesting to point out that around 60% to 80% of proteins contain two or more domains. Of course, not all of these will be amenable to this approach. In some of them, the domains won't be ligandable, or they'll be fixed in relation to one another. But there are plenty of proteins with ligandable domains that are connected by sort of flexible, non-structured spaghetti string sequences. And I think these are the ones that could be worth exploring with this approach. So in proteins like these, linking ligands for different domains in the same protein could offer a new path to modulating protein surfaces. And this could lead to gain of function outcomes like ubiquitination and degradation, as we saw here, or other outcomes as well. So that just brings, I'll quickly go through the acknowledgements. The Julie group, these are the contributors to this paper. We thank Alessio. From Georg's group, the contributors here. Thank Georg as well. And from Ivan's group, these people were the ones who contributed. We thank the cryo-EM facility at Dundee and many more people who contributed in smaller ways to this project, and of course the funding bodies. And you can find the preprint here. Thank you.